Well, good evening everyone, and welcome to tonight's webinar, which is brought to you by Southern and Tropical Beef Technology Services in conjunction with the Cooperative Research Centre for Beef Genetic Technologies, commonly known as the Beef CRC. My name is Philip Mann, and I'm a Technical Officer with Tropical Beef Technology Services based in Rockhampton, Queensland, and I will be chairing tonight's webinar. Your main presenter tonight will again be Andrew Byrne, who's a Technical Officer for Southern Beef Technology Services and based in Armidale, New South Wales. I would like to extend a special welcome to Dr. John Henshall, who is available to answer questions you have regarding tonight's topic. John is a geneticist with CSIRO at the FD McMaster Lab in Armidale, where his work includes investigating the application of quantitative genetics to economically important traits of livestock with the aim of improving livestock profitability. Of particular relevance to tonight's topic, John is also the project leader with the Beef CRC, responsible for the development of the polled gene marker test for Australian cattle broods. Tonight's webinar is titled Utilising DNA to Change Type Traits, and is the fourth webinar in the Know Your Genes webinar course, which aims to provide producers with a better understanding of DNA technology and how this can be applied in their herd. Next week, we will be covering improving production traits with DNA, before finishing the webinar course by looking at a cost-benefit analysis of incorporating DNA technology into your herd. We will again have leading scientists from the Beef CRC to assist us with the remainder of the presentations.
Okay, thank you very much, Philip, and welcome everybody. So as Philip touched on in his introduction, tonight we're going to be going through and discussing the utilisation of some of the DNA tools that are available to change the type traits. So during this session we'll break periodically for questions, but the type of topics that I plan on covering is first of all going through an outline of what we actually mean by type traits and also some of the theory behind the type traits. And then we'll focus specifically on the genetics of coat colour and what DNA tests are available to assist us if we're trying to change coat colour. And then thirdly, we'll go through and have a look at the genetics of polledness, what tests are available, but with a particular focus on the test that the Beef CRC has developed in relation to horn and polled. So that's where we're going throughout tonight's session. So if we get underway, we start talking about, well, what do we actually mean by type traits? And to define that, we're really starting to talk about those traits in which there's a sharp distinction between phenotypes of animals. So in the example for coat colour, where the animal might be black or it might be red; in polledness, where the animal might be horned or it might be polled. So the traits really are where there's a sharp distinction between phenotypes, and also where there's only a few genes that we know of that are involved in the expression of those traits. So these type traits are often also referred to as qualitative traits, and they really differ from our normal quantitative traits, or the traits which really normally relate to our production type traits, where there are many genes and there's not so much a sharp distinction between different phenotypes. So in this presentation, where I'm talking to type traits, we're really going to focus on coat colour and polledness. Now, before we start talking about coat colour and polledness, we just need to go back and refresh on some concepts relating to genetics that we have covered in previous webinars. So particularly for those people that are just joining us for the first time tonight, we just need to go through some of those concepts. So the first thing we need to consider or remember is that basically the genes of our animals are contained within the animal's DNA, and the DNA is makes up the chromosomes that our animals have. So those chromosomes are within the cell nucleus that makes up every cell in the animal's body. And within the beef animal, we have 30 pairs of chromosomes. So they have 30 pairs. They inherit one of those chromosomes from their sire, one of them from the dam, and so effectively they inherit two copies of each gene, one from the sire and one from the dam. Now the gene that they inherit, the copy of the gene that they inherit from their sire and their dam, it may be the same or it may be different. And the different forms of genes, or the different forms that the genes come in, are known as alleles. And where the form of the gene that the animal inherits is the same, so they get the same form from their sire, the same one from their dam, we refer to those animals as being homozygous for that particular gene. When the two forms of the gene that they inherit are different, so they get a different one from the sire to what they get from the dam, we refer to those animals as being heterozygous for that particular gene. Now, in heterozygous animals, what we actually see expressed, so what we see in terms of the phenotype, is determined by the interaction between those different alleles that they get. So we know they've got a different form of allele. So if we're going to talk later about horned and polled. So if we say an animal got a polled allele, polled form of the gene from the sire, and a horned form of the gene, a horned allele from the dam, so they've got a horned and a polled, what will they actually express? And that really relates to how the horned and that polled allele interact together. And in this case, we know that polled is dominant over horned, so the animal will actually be polled. So we get these two other concepts of dominant versus recessive. So the form of the gene which is expressed we refer to as dominant, the one which isn't expressed we refer to as recessive. Now, we don't always have complete dominance in that case. Sometimes those genes we get a mix of or expression which is a mix of both of them, and we call that incomplete dominance. It also leads us into this concept which we refer to of carrier animals. So where an animal may be expressing one thing but actually carrying an allele for the other form of the gene. So in that, if we say that heterozygote polled animal where it's got one copy of the horned allele and one copy of the polled allele, the animal will be polled, but we will refer to it as being a carrier for the horned allele. And we often use carrier in a similar sense relating to some of our genetic defects which we talked about in last week's webinar. So they're just some basic concepts that we will refer to tonight, a lot of that terminology. So for those of us that were joining us for the first time, I think it's important that we went through those so that we don't lose you in our terminology that we use. So if we move now to focus on coat colour, first of all, and I'll put you up on your screen now, when we start talking about the genetics of coat colour, obviously our beef animals come in, I guess, almost every colour of the rainbow and almost every shade in between, and different combinations. So we don't all just have a solid coat colour. There are animals there with broken faces, belted Galloways, etc. So the genetics of coat colour are probably not that simple, and there's probably quite a few genes going on. But there has been quite a bit of research that's actually been done into trying to understand what's going on in terms of the genetics of coat colour. And particularly that's occurred in the US, where obviously they have some quite large or more reliance on crossbreeding than perhaps we do out here. So if we start looking at those genetics of coat colour and what they have discovered, or what the research is telling us at the moment, in this case I'm starting to look at not so much what the actual coat colour of the animal is, but what their underlying genetics are. And if we look at their underlying genetics, we can classify all our beef animals into three basic colours. So there are two main ones, so we can generally classify them into black and red, and then there's also the less common one more related to brown, so reddish brown to brownish black, and I'll refer to that tonight more as the brown form. So all our animals basically can be classified into those three major colours. Now that doesn't necessarily mean that that's how they're going to be expressed, because there are a number of other genes which are actually acting and interacting with those three basic colours, which affect how the animals, or what we actually see in terms of the animal's coat colour. So there's a whole range of different genes that people have hypothesised that exist, and that have been some of them which have been discovered. So there's some genes relating to spotting, and they really start to relate to, say, things like our white face and our dorsal stripes. So if we think about our Hereford animals, then we classify those as being red, but then they're not a solid red colour because there are other genes relating to the white face and dorsal stripe which interact with those genes, the red genes, and we get the white face, the characteristic white face. There are other things there that relate to whether they're spotted or have a solid coat colour. There are other genes that relate to dilution. So if we consider, I guess, that a Charolais animal, for example, the research has shown that those animals, Charolais animals, are actually homozygous red. So we classify them into the red category, but they obviously don't express a red coat because they have, or generally are considered to be homozygous white, and that relates in the cream or the white colour that's characteristic of the Charolais. There are also some other genes there relating to roning and also to brindle. So it can be quite complex, and I think it's fair to say that we probably don't know everything about coat colour that we need to know to get a full understanding of it. But tonight, for the purpose of this webinar, what we're really going to focus on are those three basic colours, and the reason being that that's really the major gene that's impacting here, and it's also the one that relates to the DNA tests that are available. So if we consider the three basic colours, where I said the red, the black, and also what I'm calling the brown, they're really controlled by the genotype of the animal at the extension locus. Now where I'm saying locus there, it's just the name given to that particular spot in the animal's DNA where this gene exists. And the forms of the gene which the animal can have, and remember they've got two different forms of the gene. They inherit one from the sire and one from the dam. There are three different forms that they've identified, which relate to, first of all, the black form of the gene, which we'll refer here to E with a superscript D there. The red form of the gene, which we refer to with a little e, and also the wild type, which I'll talk a bit more about. So wild type form of the gene, which they commonly summarise with the E and the little plus symbol. So where we actually see they're the three possible alleles they've got. What coat colour do the different animals with the different genotypes have? If we look down here, animals with that are homozygous black, so they've got two copies of the black allele, those animals will be black. In this case, black is dominant over red, so animals with a copy of the black allele and a copy of the red allele will be black, and likewise black is also dominant over the wild type, so animals which have a black and a wild type allele will also be black, although with wild type we do tend to see a little bit of incomplete dominance where you may get a bit of a red tinge and things coming through. The red animals, with red being the recessive allele, will generally be those with the two lowercase e's, so with the two red alleles, those animals will have a red coat colour. Likewise, the animals with a red and a wild type will tend to be towards the red end of the spectrum. In the animals which might be homozygous for the wild type, they are very rare, but those animals can generally be any colour, and they tend to form into that reddish brown type of area or the brindle type of situation. So they're the different genotypes which exist, and I guess in the past, if we consider if we were trying to manage coat colour, so people have been trying to manipulate, I guess generally in a lot of cases they've been trying to move towards a black coat colour, and the only way that we were able to go through and actually identify an underlying genotype of those animals was to do some progeny testing. So to actually join animals together, see what we got, and then we could speculate about what their genotype was. But importantly, there are now some DNA tests that are available that allow us to go through and actually identify what the animal's genotype is at this extension locus, or where the gene is that's controlling these underlying three colours, whether it be black, red or wild type, or the brown allele. So those tests, I guess, really allow us with the benefit of taking animals which might have a black coat colour and identifying whether they're homozygous or heterozygous black, and by identifying homozygous black animals, then we can guarantee that the alleles that they're going to pass on to their progeny are always going to be a black allele, versus if they're heterozygous black animal, they may have a black coat colour, but they may pass on a red or a wild type of allele. We may also see situations where red animals, or likewise with black animals, people may be testing to try and see what the incidence of the wild type allele is and possibly try and reduce the frequency of that within their population. So the DNA tests which are available, There are a couple. They're the first places that they're available from. The first one, the University of Queensland, are offering this test. They're calling it the red factor, or test for red factor or for dominant black, that goes in and has a look at the genotype of these animals for coat colour. Pfizer Animal Genetics offer a similar test. They're calling it GeneStar Black, going in and trying to identify homozygous black animals. And also there's several overseas labs which are offering this test, and there may be more in Australia which I haven't listed there that I'm just not aware of. So I think at last count, they're looking at the overseas labs in the US, say there were 7 or 8 different labs offering this test. Now, actual cost of this test, it's a bit hard to speculate, obviously, because it really depends on what the type of DNA is, or tissue sample that you provide, how many you're doing, and also most of the breed societies have deals in this area. So where the black-red coat colour is relevant to the breed society, most of them will have deals with the different labs. So, but as an indicative cost, we're saying they're around $30 a head. So if you're interested in doing the testing, that's roughly the kind of price that you're going to see, but there might be variation, kind of $10 either side of that. If you are interested in actually recording the kind of process that you need to do, and it's fairly similar for most of these DNA tests, but you obviously, the first point of call I'd recommend is you contact your breed society for advice, because as I say, most of them have deals and with the different labs, and they'll also, most of the recording will be done through the breed society. If your breed society doesn't have an arrangement with a different lab, then you can actually contact the DNA lab directly. So the first thing you need to do is obviously get a DNA collection kit. You then need to go and collect the DNA samples, and you can provide, like, all this DNA, different forms of samples. So the most common one that would be provided would be hair, so taken from the tail switch, but they can also do these testing from blood, tissue, semen, etc. Once you've collected your DNA samples, you just need to complete the relevant paperwork, send the DNA sample to the lab, and the lab will do the tests and then return you with the results. So that's an overview of that recording process. So what we might do now is just break and ask for any questions that people may have relating to actually using DNA for coat colour.
Yeah, thanks, Andrew. We presently only have one question, which is related to the poll test, and that will be covered in the next session. So I certainly encourage you to send in any questions that you do have related to coat colour and the theory of the type traits, genetics. But Andrew, there's no questions at the moment, so I don't know if you wanted to continue on into the whole gene test. Well, there's one question just came through. Just hang on, there's a few questions coming through now. Okay, one question we have here. Are there tests for other coat conditions, in particular the slick hair gene?
Okay, as far as I'm aware, there aren't any tests along those lines. So as far as I know, the only tests relating to coat colour at the moment are relating to this black and red. I did see through some of the US labs that they may be starting to get some tests for some of these other genes which are influencing coat colour. I think this one was one to do with the diluted gene, but certainly I don't think there's anything for the slick coat type of arrangement.
Okay, thank you. What else we got there? Oh, one question, I don't know if you can answer this one, Andrew, but red poles often have a white tail. Do you know about this gene?
No, I don't, but I suspect it's exactly the case where the animals will be homozygous red, but there'll be the other genes that will start to interact with it. So there'll be genes at other genes at other kind of spots and other genes in the animal's DNA, which will be interacting with that homozygous red to relate to the actual white tail. But I don't know specifically if they've looked into and discovered what those genes are, or they have much understanding of them.
Okay, thanks. Another question here. If cattle have a dilution gene, can that gene be bred out of the animal by using the use of a black dominant gene?
Again, black just tends to be dominant over everything else. So if they're homozygous black, then they do tend to be black. So I'm afraid I probably don't have the knowledge of exactly how all the genes interact with one another to provide a really definitive answer, but I suspect that would be exactly the case, that yes, you could start to breed towards a black coat colour simply because black tends to be dominant over all the other, well, definitely dominant over the other alleles, but black tends to dominate over some of those other genes that come into play.
Okay, Andrew, I think there's a few other questions there, but I think we might just move on just for time. Move on to the poll gene test.
Okay, thank you, Philip. And if people do have those specific, I think some of those questions are very breed specific, I'd encourage you to contact your breed society, SBTS or TBTS technical officer, to discuss those in a bit more detail specifically. Okay, so we'll move on now to the genetics of polledness, and obviously we have John Henshall in attendance tonight, who is an expert in this area. So when we start looking about polledness, I think it's probably, I think it's fair to say it's simpler than coat colour, in that we generally have 3 different phenotypes that we're talking about. So we can see there on the top left, the animal is horned, obviously, so it's growing horns. The animal on the top right is polled, and the shorthorn bull down the bottom is scurred. So when we're starting to talk about scurs, we're talking about not a horn that is not attached to the skull, so it's just loosely attached and obviously not nearly the same size. So they're the 3 different phenotypes that we see, and there's been quite a lot of work done into actually going and trying to identify the genes which influence on whether an animal's polled or not. So what we know so far from that is that, first of all, there are several different genes involved. So in a lot of cases, if you read the textbooks and things, it always makes the horn and polled to be a fairly simple genetic case with one gene and just only 2 alleles, but we know it is slightly more complex than that. But the first thing that they have gone through and identified is there is one gene relating to horn and polled that has the major impact, and that's been identified or mapped to be on chromosome 1. But there's also another gene involved, the SCRS gene. So they do, or have identified, that SCRS is a separate gene to the horn-polled one, and some studies have mapped that onto chromosome 19, but I was discussing just before this webinar with John Henshall, and he said that there were some other studies which have shown that it may not be there. So we do know, however, that there are 2 different genes in play. In our Bos indicus cattle, then there is also an African horn gene which has been speculated about. But again, a lot of the research is still showing differing things in relation to the African horn gene as to whether it exists and also where it's actually located within the animal's DNA. Now, if we focus on the polled locus, there in particular, we know there are many different forms or alleles of the gene at that polled locus, but generally we can, or for convenience, we can generally associate those alleles with either polled or horned. And when we start talking about polled or horned, as I talked about before, we can see we have several different genotypes which we can exist, but the animals are generally polled is dominant over horned. So animals which we would consider to be homozygous polled, so as per the animal there on the left, the animal would be polled. If we switch over to the animal on the right, which would be homozygous horned, we'd expect the animal to be horned. And the animals in the middle that are heterozygous polled, we would expect to be polled, with polled being dominant over horned. Now, that is slightly complicated by the SCRS gene. So the SCRS gene then interacts with the polled gene. So obviously when the animal is homozygous horned, the SCRS gene is not expressed because the animal is horned. But when there is the animal is either homozygous polled or heterozygous polled, then SCRS will come into play and start to be expressed. Now, how that is expressed is dependent on the sex of the animal. So in cases where the animal is a male, if the animal is a homozygous SCRS, so it has 2 copies of the SCRS allele, it would be expected to be SCRS. But also if it's heterozygous SCRS, so it's only got 1 copy of the SCRS allele, then we would expect the animal to also be SCRS. In females, however, it tends to be recessive, so it requires both forms of the SCRS allele before the female will actually express SCRS. So that's how we understand it at the moment, but as I say, there's still a few differing studies in relation to SCRS. So if we start looking now at the DNA tests relating to polledness, and again, I guess this is very similar to coat colour in the past. The only way we could go in and try and speculate about what the actual genotype of the animal was at the polled gene or for SCRS was to do some progeny testing. In the research they've done, they haven't actually identified, or we can't go in and test for that polled gene directly, but there are several DNA tests which evaluate the genotype at that polled locus by using a linked gene marker. So, and there are different companies offering these, and they do it slightly differently and use slightly different gene markers. But the important point there that I should stress is we're not actually going in and testing for the polled gene directly. We're just looking at a marker. And that causes us some issues with accuracies we'll talk about because we're not actually looking at the gene itself. We don't actually have a gene marker at the moment for SCRS, so we're just purely looking at what we're talking in the horn-polled gene. Now, different companies which are offering these tests, I'll start with the 2 overseas ones which are looking at the bottom. Those tests have been available for longer overseas than what some of our more recent tests are in Australia. So there's a test offered by Identity in the US for around $55 a head, and it's a breed-specific test. So you can see there it's only available for Charolais, Gelbvieh, Hereford, Limousin, Simmental, and Shorthorn. MMI or Metamorphics are also offering a test for around $110 US for Charolais, Gelbvieh, Hereford, Limousin, Slairs, and Simmental. Now, both of those tests, as I understand it, certainly the Identity test, for those that were involved in the first couple of webinars, they are SNP-based. So the gene markers that they're looking at are SNPs, or single nucleotide polymorphisms, rather than microsatellites. More recently, we've got tests now that are available in Australia. Both the tests, the companies which are going to be offering these tests, the University of Queensland and Pfizer Animal Genetics, are offering the test which has been developed by the Beef CRC, which we'll go on and talk about in a bit more detail now. The University of Queensland are offering it for kind of, again, an indicative cost of around $33 a head, but it's cheaper if you're going through your breed societies. Pfizer Animal Genetics are planning on releasing this test. They haven't yet released it, and they haven't got a time frame on it yet, but it's likely to be shortly, and they haven't got a price available on that just yet. So they're just an indication of where you can get the testing done. Obviously, I guess traditionally you could get it for those breeds in the US, but we now have the Beef CRC developed test which is being released through labs in Australia. However, the reliability of the Beef CRC test, as we say, is more suited or more accurate in different breeds. So it's not necessarily suited to all breeds. But we will expand on that. So I guess before we start looking at the Beef CRC test, I think it's important that we sit there and say, well, why would you do a DNA test for polledness? And just to explain that, I guess the reason that you'd start to consider doing DNA tests for polledness, in my view, would be if you're trying to dehorn your animals. So obviously, if you were trying to breed horned animals, then the animals, there'd be no need to do a DNA test because they're homozygous horned, and so there'd be no need for the testing. Where the test really starts to come into be use to us if we're trying to dehorn our animals genetically, so we're trying to breed for polled animals, and we're trying to differentiate between animals which are homozygous polled or heterozygous polled. And just to show you the effect of that, I've got two different scenarios up here on the screen. In the first case, we'll say we're a horned herd, and I've used Hereford pictures, so I'll consider it to be we're a horned Hereford herd, but it would be exactly the same across the different breeds. And so we've got the females over on the side here, and we're going to put a polled bull over them. Now, if we were just using any polled bulls, then it'd be a bit of a lottery as to whether that animal would be homozygous polled or heterozygous polled. But if we used a homozygous polled animal, then as per the example on the left there, then we would expect that 100% of calves would actually be polled, and some of them will be SCRs, so that the SCRs gene would come into them. But if we look at the genotype, they would all be heterozygote polled, and so on average we'd expect them to be polled and SCRed. I guess what we could say is that we wouldn't expect any horned progeny. However, if we used a heterozygote polled animal over those animals, then we'd expect 50% of the calves would be heterozygote polled and polled or SCRed, versus 50% of calves would still be horned. So just by identifying, if we had a horned herd and we're trying to take the horns off them through the genetics, by identifying the homozygous polled bull, then we get a lot more bang for our buck. If we start considering the next generation, so and we identify the animal the bull we're going to use is homozygous polled versus heterozygote. So now we've gone one generation down. So we'll say we've used a homozygous polled bull. We come down here. All our progeny are obviously heterozygote polled. If we put a homozygous polled bull over them, as per this example on the left, we would expect that 100% of those calves again would be polled, or some of them may be SCRed, but 50% of those would actually be now homozygous polled animals, and 50% of them would be heterozygous polled. If we look over on the right, however, if we put just a polled bull over them that happened to be heterozygous polled, then again 25% of those calves would be actually homozygous polled. 50% of the calves would be heterozygous polled on average, so we'd expect them to be polled or SCRed, and 25% would still be horned. So I think those two examples just show us if we're trying to take the horns off our animals by genetics, by using this DNA test to identify homozygous polled bulls and use homozygous polled bulls over our females, or likewise on the female side of things, identify homozygous polled females, we can get a lot further than what we would if we're just relying on the phenotype. So if we focus now on, well, let's understand the test that's available a bit more, and in particular the beef CRC test which has been developed. To give you some background, the beef CRC started to look at this, developing a test particularly for Bos indicus cattle. If you consider those overseas tests which I said were available before, they weren't available or applicable in Bos indicus cattle, and with the high Bos indicus population within the Australian beef herd, the CRC went in and started to look at whether they could develop a test specifically for Bos indicus, and so most of the early research was done in Brahman animals. As per the other tests, we're not actually going through and identifying the polled gene, but we're looking at the genotype of the animal at a gene marker, if you like, which is at a linked marker which is closely linked to the polled gene. And what that means, in essence, is that generally those two genes, both the marker and the actual gene affecting polled, tend to be inherited together. So when we go through and identify at the marker locus, there are many different alleles present, but the research has shown that some of those alleles are always associated with polled, some of those alleles are always associated with horned. So we can do the test. We're not testing for the gene directly, but we're looking at the association between the genotype of the allele at the marker locus, and therefore we can infer what that genotype is likely to be for the actual horned polled gene. Now, unfortunately, some of the alleles which they've identified tend to be associated with both polled and horned alleles at the horned polled locus. And in this case, we're referring those as ambiguous alleles because they don't actually tell us anything about what the genotype of the animal is likely to be at the horn-polled. And the critical thing to understand is that these ambiguous alleles do exist. The frequency of them differs, however, quite significantly between breeds, but how frequent they are really determines how useful the tests are. So in some breeds where they're not that frequent, and most of the alleles, or the majority of alleles which are present, we can confidently assign to either be polled or to horned, the test is very, very useful. But in other breeds where the ambiguous alleles are quite high, or the frequency of them is quite high, then the test isn't quite as useful because there's a lot of cases where we can't actually determine confidently what the genotype of that animal is likely to be at the horn-polled locus. We do also have some inconsistencies, so we're discovering as the research goes on that the horn-polled situation, there might be some other subtleties going on in relation to the genetics that we're not yet aware of, and that can lead to some inconsistencies in the phenotype. So if we use the CRC test, and we identify, for example, the animal is homozygous horned, but the animal is actually expressing a polled phenotype. So there are some little subtleties in there, in a small number of cases, that is causing the test not to be as perfect as we would hope for. So to actually validate the test and show you how useful it is, there's been quite a bit of work done by the beef CRC. There are three different major parts of the validation work which has been conducted, and I've just put some statistics up on the screen there to give you an idea of the different numbers of animals that are involved by breed. So the first bit of work which was done was obviously in the experimental population, so when the CRC were first going through and trying to discover this gene marker, and as I said, it was initially focused on developing a gene marker for Bos indicus cattle, so there was quite high numbers of Brahmins and Santa Gertrudis in the experimental population. The CRC then, though, once they discovered that gene marker for those breeds, then did roll it across and try and test it in other breeds, a number of other breeds, and see if it was also working in those, so that we weren't just limited to these Bos indicus animals. So the first lot of stats there are to do with that experimental population. There was then a major project run last year where they were trying to develop a database of DNA for well-used high-accuracy EBV industry sires across the different breeds. So there was a call that went out to members to try and submit any semen, and I think 2 semen straws on particularly well-used sires. All those animals that came in were then tested by the CRC for the horn-polled marker. So you can see that the summary of the number of animals there that came in. Not all those industry sires, were tested, but those numbers there were. And then we've also got the third component of information which has come in, which has actually been submitted by breeders. So the University of Queensland has been offering this test now for 12 months or so, and a number of breeders in the different breeds have actually sent samples in directly to the University of Queensland to do some testing. So they're the numbers which are evolved, and you can also see the different breeds. I guess if you're a member of a breed that isn't listed there, then all we can say at the moment is we're not really sure whether the beef CRC test is applicable in your breed or not, and we don't know how accurate it would be. But obviously if you collect some samples and we can collate them and send them in, then we do have some options of starting to validate it in your breed. So how accurate has the test shown to be? And this next table here on your screen will give you an indication in the different breeds. I should say there has been quite a lot of validation results released, and I think one of the pluses from a beef CRC point of view is that we do have full access to the validation results, which really allows us to make an informed decision, and that's, I think, very critical to the future work with DNA, in that we do have good validation information available. So I'd encourage you, if you're looking at the horn-polled test, to view the full results on the beef CRC website. So I've got an address up there on the screen, and there is a full report available from that site, which will really break it all down for you. I have plucked some information and put it on this slide. So we're looking now at samples which were submitted across the 3 different experimental populations that had a polled phenotype. So the animals themselves were polled, And what we have here is on the left-hand side here we have the different breeds, and across the top we have the different genotypes which were identified using the beef CRC test. So the first column was the number of animals, so that remembering they've got a polled phenotype that we would consider to be homozygous polled. The next column is those that were heterozygous polled. The next ones are the ones that the test actually identified were homozygous horned. Then we have three columns, one that had one polled allele and another ambiguous allele. The next ones were horned with an ambiguous allele, and the last column is those that have two ambiguous alleles. So as we say, in terms of when they were validating this to see how useful the test was, then they're really looking at, the first thing was the frequency of ambiguous alleles, and you can see that differed very much across the different breeds. So particularly in Brahman, which is probably what we'd expect given a lot of the early research was done in Brahman, there was not a very high percentage or frequency of ambiguous alleles. So the test was really good from that facet in Brahmins. In other breeds, however, the percentage of ambiguous alleles was quite high. So for instance in Limousin we have there at 61% of alleles were actually ambiguous, or percentage of animals actually had an ambiguous allele. So now that just limits, I guess, in each of the different breeds how frequent the ambiguous alleles are is to, I guess, how many animals that you send in you'll get a definitive genotype on. The second thing that we can look at is, of the actual genotype, did it make sense with the phenotype of the animal? And the last column I've got there is just the percentage of times where the actual genotype which came up was inconsistent with the phenotype that had been recorded for the animal. Now we have no guarantee in a lot of this work that the phenotype which had actually been recorded for the animal was accurate. So a possible source of that error there may actually be in the phenotype recording, or we may have some other allele or other genes at play that we're not quite aware of. So you can see there, I won't go into each individual breed, but if you do look at the information on the Beef CRC website, it will give a full, I guess, explanation of what the different results in each of the different breeds are meaning. So some take-home messages, though, from the validation work which has been conducted. I think it's important to note that the Beef CRC test, first of all, is not looking at the polled gene directly, but it's looking at a marker, and that marker is not perfect. But having said that, either are the overseas tests which have been developed which were SNP-based. So they had a different level of accuracy in the different breeds. The important thing, though, however, is that this test is working very well in Brahman. So if you're a Brahman breeder out there, then I think that test is going to be very, very useful to you. It's also showing a lot of promise in some other breeds, so particularly, for instance, Hereford. If we looked on that previous slide, the percentage of ambiguous alleles wasn't that high, and it's showing that it could be quite a useful test for you. In some other breeds, we probably need some more validation work. Some of the numbers are quite low that we have available, but it is showing some promise. And I guess if you consider it, say in the Limousin breed, where I think we had around 60% ambiguous alleles, 60% of cases, it might be that it's still worthwhile test to screen a whole lot of animals. That just means that you won't get a definitive result on a significant proportion of those. But it may still enable you to really definitely identify those animals which are homozygous polled, and really focus on using those in association with obviously your selection for the other economically important traits. What we do have coming out of that validation work is some changes in the way that the results were being reported. So I guess the expectation when the test was being developed was it would be able to report a definitive genotype, so either the animal's homozygous horned, homozygous polled, heterozygous polled, or homozygous horned. What now the test is coming out and doing is generating the probability for each particular genotype. So in the example I've got there, it's looking at the alleles the animal has to coming up and identifying what the probability is the animal's homozygous polled, probability it's heterozygous polled, or the probability which is homozygous horned. Now that's what the test is doing. there's a lot of discussion at the moment as to how those results are reported. The University of Queensland are now starting to report just the probability that the animal is homozygous polled. So given that the major use of the test is hopefully to look at polled animals and identify whether they're homozygous polled or heterozygous polled, so you'll start to see, like the example there on the screen, just a simple result for each animal, giving an indication in this case that there's a 90% chance that that animal is homozygous polled. And I think that represents a really big change in mindset that we need to get our heads around when we start looking at this test, that we're now not giving a definitive genotype, but we have a tool which will give us the probability that an animal is homozygous polled, and that there is some degree of risk there or error that we need to manage. In the case of Pfizer, they haven't exactly released their test yet, but from talking to them over the last couple of days, they are probably going to report their results in a slightly different manner. So they are planning on picking, I guess, the most likely genotype and giving you the probability of being that genotype. So in the example above, it would probably be exactly the same display as University of Queensland. They'd report that animal as being homozygous polled with a 91% probability. But there will be cases where they'll report the probability that it's heterozygous polled if that's the most likely one. So if you're interested in testing with the horn polled test, then the process is exactly the same as for the horn poll, the coat colour. You should contact your breed society as a first point of call and see whether they have an arrangement with, at this stage, the University of Queensland, but there may be other breed societies now starting to look at arrangements with Pfizer as they plan to release the test. Contact your breed society, or if the breed society doesn't have an arrangement, you contact the DNA lab directly. Get a DNA collection kit, go out, collect your DNA samples. Again, hair is perfectly adequate, but you have the option of submitting other forms of DNA across blood, tissue or semen. Collect your samples, fill in the required forms, send those off to the DNA lab, and then the results will come back to you. So that's the general recording process. One thing I didn't mention throughout that presentation is there is some further validation work being done. So the AMLA have just put some more funding up to go in and do some further validation, particularly in some of those other breeds that we don't have a full set of results from because we had low numbers. So there'll be further work done over the coming months towards the end of the year to give us updated validation results, if you like, or updated ideas as to how useful the test is in each particular breed. So hopefully that's given you a bit of an overview. We'll now break for questions, and I'd certainly encourage you to utilise the services of John Henshall tonight.
Thanks, Andrew. Yeah, we've got a few questions there. But, as Andrew said, yeah, certainly send in those questions and get John to answer them, take the opportunity for John to answer them. Firstly, John, there's a few questions there about SCRS and the SCRS gene. And one question we have is, will we see a genetic test for SCRS? Andrew did mention that we currently don't have one, but will there be one in the future, and when do you think that will be? John, can you...
Thank you, Philip. Thank you, Andrew. Yeah. Yes, can you hear me now?
Yes, that's fine. Go ahead, John.
Thank you, Philip. Thank you, Andrew. I guess the answer is maybe we'll have a test for SCRS. At the moment, what we're finding is that in some breeds, SCRS are very, very common in heterozygotes, in animals that are heterozygous at the polled locus, and that's certainly the case in Brahman and in the Belmont bred animals we've looked at. And in other breeds, we almost never see SCRS, and so we do know that it is a genetic. There is a genetic cause to SCRS, but just because it's genetic doesn't necessarily mean that it's due to a single gene. And if it is due to a single gene, then I think that fairly soon we will find a test for it, and we would be able to market that. But if it turns out that SCRS is due to lots of genes, just like other traits, quantitative traits like size and weight and growth traits are due to lots of genes. If it turns out that skers are due to lots of genes, then it's probably unlikely that we'll ever have a test for it. So the answer is it depends on the mechanism. If it's simple, just like polled and horned is simple, then yes. But if it's really a quantitative trait, then probably not.
Okay, thanks, John. This probably leads into the next question, or a couple of questions. And how does the skers fit into the phylogenetic makeup of an animal? Is it more associated with heterozygous polled, and can it be associated with homozygous polled animals?
In the data that we have, in the breeds that we've looked at, where skers are common, they're overwhelmingly occurring in heterozygous animals. In Brahman, the frequency of skers in heterozygous animals is up around 50%, and the remaining animals are either polled or horned. So you could say that in Brahman, almost no animals that aren't heterozygous do have skers. So the presence of skers is actually a pretty good, if you see skers, then you can be reasonably sure, at least in that breed, that you've also got an animal that's heterozygous for polled. The problem is that there's lots of heterozygous animals for polled that don't have skers. So it's not, well, it's not a very good diagnostic from that respect. But certainly we have very few animals in the breeds that we've looked at where there are skers, or the animals have been reported as skers, and they are other than heterozygous at the polled locus. Okay, thank you. I guess there's another question related to that, the scared cattle, is if they are polled and not scared, are they then homozygous polled? Unfortunately, not necessarily. It would be very nice if skers was a perfect test, but in none of the breeds that we have is the absence of skers a good indicator that the animal is homozygous polled. Okay, we've mentioned, well, Andrew mentioned that the polled gene test is a linked marker. Is it realistic to think that the polled gene may be identified rather than a linked marker to improve the accuracy of the gene test? I think it will be identified, assuming that it's something fairly simple. People now are, if you look at the way the technology is moving, people are now able to sequence animals fairly cheaply, and this is to actually, instead of just looking at markers along the genome, to actually look at the bits and pieces that make up the genome on individual animals. So certainly we're interested in pursuing that here in Australia, and I'm sure that lots of the American groups are as well, and European too. My main reservation would be that if it is something really simple, there would have been a pretty good chance that we would have found it by now, because we've found the cause of lots of other things that are less easy to look for than polled and horned. So in the back of my mind there's a thought that, well, maybe it's a little bit more complicated than we think, so perhaps that direct test might be a bit further away than we think. Okay, thank you. Andrew also mentioned the African horn gene, and the question is there African horn gene in the British breeds or Bos Taurus animals? The African horn gene story really starts from when people started to bring Sanger animals, so African Bos Taurus animals, into Australia, and sometimes we saw, or people observed, what they didn't expect when it came to horns in progeny that they expected not to be horned, and I guess again, a bit like skers, the fact that something is genetic, and it's definitely a genetic effect, doesn't necessarily mean that it's due to a single gene. So the African horned gene is really a hypothesis rather than a gene, I would say at the moment. It's a plausible explanation for something that didn't seem to fit the pattern that we expect with British breed cattle. But I think that the evidence is still, I don't know that the evidence is really strong enough yet for us to say that it's definitely due to a single gene. And in terms of as to whether or not it occurs in British breed cattle or in other Bos taurus animals, I don't think that I've seen any data that would make me have an opinion either way on that.
Okay, thank you. We have another question here, again going back to the link marker of the poll gene test. In relation to the gene markers that have been identified for tenderness in Bos index cattle, are they based on linked markers similar to the ones that are in the CRC test for polledness?
I probably can't give you an answer as to all of them. Certainly some of them are definitely going to be due to link markers, but there may also be some out there that aren't due to link markers, and I'm not an expert, and I'd really want to check before I said yes or no to that. But both are possible, and both might be quite good tests. Just because something's a linked marker test doesn't mean that it's not a good test. But I don't know the answer to that, sorry.
Okay, thanks, John. I guess there's a question here in regards to the poll tests currently being offered breed specific, or are they likely to be in the future? Perhaps you could just comment on that, John, given the fact that the test may be not working as well in some breeds as it is in others.
Well, in a way, our test is also breed specific because which alleles are ambiguous depends on the breed. Some of an allele that in Brahman is very clearly associated with horned, in another breed might be ambiguous. So in a way, we said, just as we're not prepared to say our test works or doesn't work in a breed that we've never seen any data from, likewise we really do need to know the breed of the animal before we can give a good prediction with our test as it is. So it is in a way also breed specific. I think that if we ever get to either the causal mutation, the thing that really causes this, or something much closer to it, then we might have something that works across breeds, and certainly that would be everyone's goal, but at the moment, while we're still using linked markers, then all tests almost are likely to at least be to some extent conditional on the breed.
Okay, thank you. Another question we have just within the breed. So could different populations within the same breed have different results in terms of the or frequency of ambiguous genes or ambiguous alleles?
That's actually a really good question, and it's one of the things that we're working on now, because if you think of a breed as a population, and there are within breeds subpopulations, if we can identify that in some branches or some bloodlines or some subsets of various breeds, something that over the whole breed is ambiguous might actually be quite a good predictor in some families or in the progeny from some studs, then we'd be able to improve our test and our current test to perhaps and get rid of the ambiguity associated with some of these breeds. And you could even take that a step further and say that, well, if we looked at the whole pedigree of all of the animals, then perhaps something that just on the test alone might be ambiguous. But if we know the sire, the grandsire and great-grandsire, and also the genotypes and phenotypes of cousins, nephews and other animals in the pedigree, perhaps that additional breed structural information or pedigree information might allow us to give fairly good predictions, even though the test on its own is still giving an ambiguous result. So certainly the answer to that is yes. You'd expect where there are ambiguous alleles, then they'll be in certain branches of the pedigree, and if we can get an understanding of the genetics, the inheritance of polled and horns in that branch of the pedigree, then maybe those alleles are no longer ambiguous.
Okay, thanks, John. Another question we have here is asking how many crosses from a horned animal to a polled animal would you estimate for the poll to become homozygous when using a homozygous polled bull? The second part of the question is it common to lose muscle when going from a horned to a polled animal?
So the first part of the question is really about, as I understand it, is about how many generations do you need to keep breeding before you've got rid of the last of those homozygous or heterozygous animals, the carriers that you can't see. Well, if you don't have a test, then that can take a very, very long time. But if you do have a test, then it needn't be very long at all. As long as you, once you've, if you're consistently using homozygous polled bulls, then really within a fairly small number of generations, all of the cows are going to be, well, the first generation's heterozygous, and then within a few generations, a very high proportion homozygous polled. It's really where the test is valued. The second part of the question, you'll have to remind me, sorry.
The second part of the question was, when you're going from a horn to a polled animal, are you going to lose any muscling in the animal?
Yeah, that's also a really good and a very important question. What it comes down to is it depends on where you're getting the polled genes from. If you have a horned cow, a horned herd, and you buy a polled bull, then the muscle is really going to be based on the quality of that bull and his muscling. So if you're buying breed plan bulls and this is a polled bull with good EBVs for muscling or whatever other traits you're interested in, then you shouldn't lose anything at all. But if you go and buy something just because it's polled and it has fairly bad EBVs for other traits of importance, then potentially you could suffer a cost in muscling or whatever other traits you're interested in. So it really is important when you're looking to source, if you're moving to polled, and then to find those bulls that suit the breeding objective for the rest of your, whatever your current breeding objective is, and there's nothing intrinsically linking the absence of horns to muscle traits. There's no data on anything, any adverse associations there. But that doesn't mean that you should ignore the EBVs for these other traits that are important when you're choosing a bull.
Very good, thanks, John. We'll just probably take a couple more questions because time's getting away on us, but just in relation back to skers again, identification of skers. Skers are supposed not to be attached to the skull. How can we be sure of this? Can they be small and attached, and therefore they are small horns, or can they be identified as skers?
It's probably, I'm not the best person to talk to about that. I've probably seen many fewer skers than most of the people who are in the audience, so I'm definitely not an expert here. I take the view that from a genetics perspective, in terms of the test that we have, the range of things that people might be reporting as skers is possibly quite large, and it may differ between breeds. But despite that, what people are reporting as skers are almost always in heterozygous animals.
Okay, thanks, John. Probably take us to our last question now, just relating to the reporting of the genotypes of the whole gene test. We have a question here. We had a DNA test done for Polled by UQ. The sire was a heterozygous Polled over homozygous Horn. One result was homozygous Polled at PP 4.75%, pH 90.5%, and homozygous Horn 4.75%. Another animal came at 2.5% homozygous Polled, 50% heterozygous Polled, and 47.5% homozygous Horn. How do we interpret these results? Both offspring are visually polled, and these are from a mini Hereford breed.
So in coming up with those results, we would not have taken into account the fact that the animal was phenotypically polled. We would have based the test results just on the DNA test that came back. So in the first case there, where the animal was, the probability was overwhelming that it was a heterozygous polled animal, that would appear to be certainly the most likely phenotype. In the second, genotype, in the second case where it's allocating the animal 50% to being heterozygous and 50% more or less homozygous horned, knowing that the animal is polled phenotypically means that that second possibility, homozygous horned, is most unlikely. So it's much, much more likely that the animal is also a heterozygous, one horned allele, one polled allele. But in the test results, we're basing it just on the DNA, not on the fact that the animal also has a phenotype. The animal is, you can observe that the animal has no horns. So you might, in that circumstances, say that if you put there's a very, very strong probability that both of those animals are heterozygous.
Okay, thanks very much, John. Look, I think that's pretty much covered all the questions we have, and we're running out of time a bit, so we might just pull it up there and move on with the wrapping up the presentation. So thanks very much for that, John.